Brilliant. Should we make a start then? So, hi everyone, and welcome to the TIA Centre Seminar. So, my name is Adam, and I'm a research fellow here at Warwick. And for those of you that are new to our seminars, in this seminars, we aim to invite researchers from across the globe to present their recent work. And we are sure others will also find it informative and interesting. So, to get started, I'd like to introduce James Diao, and he's from Boston in the US. James is a fourth-year MD student at the Harvard MIT program in Health Sciences and Technology, supported by the Paul and Daisy Soros Fellowship. His research explores how clinical, genetic, and computational tools can improve health for diverse populations. Previously, he developed algorithms predicting molecular features from cancer histopathology images at PathAI, and for predicting cardiovascular fitness from wearable devices at Apple. He graduated from Yale University with degrees in statistics and data science, as well as molecular biophysics and biochemistry. And the title of James's talk today is Human-interpretable image features derived from densely mapped cancer pathology slides predict diverse molecular phenotypes. So James, if you're ready, then please feel free to share your slides. Let me just make sure you can. I've made you a host, so you should be able to share your slides now.
Okay. Let's see. Right. Oh, starting halfway through the talk. Okay. Can everyone see my slides?
Yeah, we can see those. Okay, thank you.
All right. So hello, as you mentioned, I'm James. This work I'm very excited to talk with you about today is mostly going to focus on this paper that we published in Nature Communications about sometime last year, 2021, titled the article, same title as this talk, co-led with my classmates, Jason and Wan Feng. And this was performed as my MD thesis in collaboration with some really great folks at PathAI, where I worked for about 2 years. And so that's why most of the co-authors listed here contributing to the work will be from PathAI, and why all the code performing the downstream analyses is hosted there on their GitHub. So my disclosures, I think mentioned earlier, so obviously with PathAI, and then perhaps less relatedly with Apple, but nothing else to disclose. So let's get into it. So I think for this crowd, probably won't need to spend too much time on background, but I'll just very quickly breeze through some of the motivation here. So as you know, the analysis of histopathology slides, the gold standard, still remains an inspection by a trained clinician who will then observe the structures, patterns, and morphology to assign certain interpretations, sometimes with the assistance of special stains or other methods. But the goal at the end of the day is to assign potentially something like diagnosis or prognosis. And with the advent of modern computer vision methods, we've gotten the ability to, with or without annotations at the slide level or smaller biological entities, to be able to use machine learning methods to supplement some of these tasks that pathologists are able to do, or go beyond those and start tackling tasks that are currently not able to be performed by pathologists today. And then beyond just the analysis of pathology itself, the thought is that combining this information with genetics, radiology, and other clinical data will all help enable a more fine-grained understanding of disease in order to personalize treatment and to provide better care for patients with cancer and with other diseases that have this type of histopathological data. So the main question that's motivating our specific query here is, can we represent histology slides with human-interpretable features in ways that offer advantages over direct end-to-end models? And the reason we're interested is because I think the advantages of end-to-end models are very clear in that when you're directly optimizing for a certain objective function, you're able to take advantage of signals and hidden correlations that might not be known and don't require any explicit engineering, and have resulted in some of the best performance in recent years. But this also comes with some disadvantages that human interpretable features might be able to counterbalance. So some advantages of this include improved interpretability, which is especially helpful for building trust and reliability within clinical settings where being able to justify decisions is paramount. And of course, I'll talk a little later about comparison with other interpretability methods that are used for intent models like class activation maps or saliency maps. Another advantage is being able to quantifiably and scalably connect the predictions directly to biological entities, which is potentially helpful for future investigation. And then it also kind of defines this hypothesis space of areas that the machine is allowed to learn, which enforces an inductive bias that on the one hand limits overfitting, which can be good, can sort of get in the way of the machine cleverly learning spurious associations that might not generalize, but also, as you know, can potentially lead to underfitting as well. So it does enforce this inductive bias. And then just a little bit of context about previous work on HIFs. And so I haven't been able to do a thorough scouring of the literature since this was published, so maybe this is not entirely up to date, but when we were doing this work, we were looking at some of the other work that had been done in this space. And our impression was that a lot of the previous work was often on one tissue or cell type. So for example, something like looking at lymphocytes or plasma cells or macrophages, and how well they associate with things like prognosis. They often rely on extensive downsampling of the image in order to make it computationally tractable, and are often specific to a single cancer or outcome. And we saw the most common by far was lung adenocarcinoma or lung squamous cell carcinoma, partially owing just to the really large number of samples in the public domain relative to a lot of other cancer types. And then the easy accessibility of certain outcomes like progression-free survival, for example, as another target that was often predicted, but usually it was just one rather than multiple things. So we think major contributions that we aim to deliver with this paper were to, for the first time, start to investigate combined cell and tissue properties. So not just how many lymphocytes or plasma cells are enriched in the tissue, but where they are. So stromal lymphocytes are very different from tumor infiltrating lymphocytes. And so to be able to explore that more thoroughly, to be able to do this using exhaustive mapping of the cancer histology slides at subcellular resolution. So meaning having a prediction for which tissue class this is at every 4 microns and which cell is present, if any, at every 2 microns is something that we thought would enable an unprecedented level of exploration of the tumor microenvironment. And then also just to explore the limits to which these features can generalize across different tasks in different settings, trying to validate the performance across 5 different molecular signatures and 5 cancer types. So this is what we set out to do. So this is the overview. There's no need to look into it really closely because this slide will come up multiple times throughout the presentation, but just to focus on step 1, which is, you have all these slides and you have to train deep learning models to obtain the tissue and cell specific spatial information before you can do anything with it. So I'll talk first about how we went about doing that. But maybe I'll pause there if there's any questions really quick before I jump into step 1.
I don't think there's any questions yet, so feel free to carry on.
Excellent.
Yeah, feel free to carry on going. Thank you.
Great. I will move to step 1. So a quick overview of the data. So this study was using the Cancer Genome Atlas. So looking specifically at these 5 cancer types shown in the columns above. So these include BRCA, breast adenocarcinoma, LUAD, lung adenocarcinoma, LUSC, which is lung squamous cell carcinoma, SKCM, skin cutaneous melanoma, and STAD, which is stomach adenocarcinoma. And you can see there are the number of slides range from, so WSI is full slide image, ranges from in the mid hundreds to over 1,000 in BRCA. The number of patients is slightly lower because some patients have more than one slide attached to them. And then, so later I'll talk about how we choose which slide to analyze in the downstream analyses where it needs to be one patient per sample. And then we have, as you can see, a very large number of cell level annotations, about 300,000 across these 3,000 slides or so. And then a smaller number of tissue level annotations. And then these data, besides the imaging themselves, also have case match clinical and sequencing data, and a lot of intermediate data files that other groups have analyzed and produced based on these foundational data. So things like processed, immune signature data, or like deconvolutions that have been performed. So I'll talk a little more about how we access those data later. The other source of data that we use is PathAI's partner data sets that it acquires from the different biotech partners that it works with. So you can see this adds a substantial number of slides and over 1 million cell level annotations and over almost 150,000 tissue level annotations. So we use this just to supplement the training procedure in order to help make sure that this generalizes beyond just the TCGA and any batch effects that might arise from uniform processing. So model development. This is a very standard workflow. I'll go through it pretty quickly. I'm sure many of you are familiar. So we obtain.
Sorry, we just got a quick question from the audience here.
Yeah.
Do you mind if we?
Yeah. Hey, James. So can you go? Do you have any information on data? So these annotations, in particular, in your next slides. Like, what are the kind of annotations you did on cell level? Like, is it segmentation or just point annotation? What are the classes?
Yeah. So let's see. Yeah. So the classes are
If
here.
you have, I'm not in a rush. If you don't, I would appreciate it if you explain it now. But if you have some slides, I can wait.
Absolutely. Does this slide here answer your question about the different classes, and then I can tell you about?
Oh yeah. Yeah.
Okay. All right. Yeah. I'll go through the slide, and then if I don't answer your question, definitely just ping in, jump in again.
Thank you. Cheers.
Yes.
Thank you.
Cheers. So yeah. So step one is obtaining the annotations. So there's two types of annotations. There's the regional tissue annotations, and these are annotated by polygonal selection. So they'll click, and then they have to click back to the original point, and then the convex hull of those points ends up being the selection area, and then that will be a single annotation. And then during training, that region will then be sampled multiple times to produce patches that will then become positive and negative training examples. And then the labels that are used here are cancer epithelium, cancer-associated stroma, necrosis, and then normal tissue or background. And then for cells, this is just click in the middle of the cell, single click. And this includes cell types like lymphocytes, plasma cells, macrophages. You can read the rest of the list, but essentially 5 major notable cell types, and then there's just the other background non-cell category. And so very standard procedure, splitting the data set, data augmentation procedures to enforce certain invariances into the model training process, including to rotation, brightness, and saturation. Oh, and just as mentioned earlier, there will be many different annotations per slide, many patches drawn from each tissue regional annotation. And so you end up with up to thousands of training or validation, either training or validation patches per slide. And each slide is only one or the other. In fact, each tissue source site is only one or the other. It's segmented at the level of the source sites. And then I'll talk more about the process by which this is done, but we use nested cross-validation to tune the model hyperparameters and architecture. And we aimed primarily for maximizing accuracy in the validation set, or each validation set. And so here are some of the things that we were searching along to optimize the model. Yeah, and so one thing that we want to emphasize is in this process, we tried to have significant pathologist involvement because we didn't want to over-optimize against just metrics or summary statistics that might not properly capture whether the model is accurately representing the biology. And so so first we collect primary annotations where they're just asked to go and click on all the lymphocytes they can find in like 10 different patches in the slide, or find 10 different patches where they can draw a polygon. So this is not actually an example of the annotation process. This is just a picture I found online to go along with it, because I don't have access to the slides platform anymore, and so couldn't attach a proper slide. Oh, and I should say anything that's not referenced is just from us, and then anything that's referenced I got from somewhere else. And so then we would train models. It's a ResNet inspired CNN architecture, and so you can sort of see, yeah, it's just very standard model training. I won't go into that. And then we'll sit down with the pathologist and look specifically at if there's any systematic errors. So for example, on the left in the heat maps, you can sort of see as you go from left to right, improvement of several big errors. So one of them is when it sees like little flecks of tissue sort of free floating out at the edge of the sample. It at the beginning of training will often mistake this as necrosis. And so this is something that our pathologists will easily identify, point out to us, and then we'll collect targeted annotations specifically against things like that. And then, or things like, near the edge of the tissue, there's decreased confidence in being able to assign cancer epithelium versus stroma. And so being able to find where the model is getting most confused, where things are being undercalled or overcalled. And so we look at the confusion matrices and the false positive and false negative rates to handle that. But we rely primarily on the pathologists letting us know whether they really believe that like the downstream summary statistics, like the number of lymphocytes within 80 microns of the cancer stroma interface, whether they would actually believe that that's associated with a real thing. And so and then we go back, and when we collect annotations again, it'll be targeted against what we're doing poorly at. So macrophages have been a huge pain in our neck during this process because they're pretty difficult to learn, and even pathologists have trouble sometimes confidently telling what our macrophages are and aren't. So then we'll send out jobs specifically with more macrophages, or have them not just label macrophages, but find the mislabeled ones and correct them. And so that with each subsequent round, ideally, the annotations are becoming harder the more and more difficult cases, the ones where the model's getting wrong, rather than the ones that it's nailing on its first try.
Brilliant. Can we ask you a couple of questions here, please? So, Mr. What was your question?
This is my question. I just want to make sure. So this pathologist in the loop is just in the annotation acquisition step. You didn't get pathologist confirmation on all the samples in this study, right? So it's just a regions annotation.
Sorry, can you repeat the question?
So I'm asking, this pathologist in the loop process, which is really nice, did you do it when, so we are still talking about annotation curation, right? So you're not talking about when you did the prediction on the whole slide images, on all the cancer whole slide images, you know what I mean? Is this only about the...
This is just for creating ground truth annotations.
Annotations, okay. So
that's what you're asking, isn't it?
Yeah.
Yeah.
Is this only for ground truth annotations? The pathologist in the loop process?
It's also for assessing the model itself. So we get annotations, and we get annotations, we train a model, we show the heat maps to the pathologist. The pathologist tells us where we're messing up the most and where it needs the most work. And then we'll send out an assignment to the pathologist network, which is a different set of pathologists, specifically targeting those weaknesses, and then train new models, talk to our pathologists again, and then send out more correction annotations, and then keep going until they are satisfied that what they're seeing is an accurate representation of the tissues and cells.
Okay, makes sense. But still, I'm a bit confused. I'm sorry. But it has been done on all the slides, like on whole slide level processes.
So we produce the heat maps on every single slide, but the pathologist doesn't look at every slide. They sort of scroll through the thumbnails, and then they look for things that stand out to them, and then maybe they'll like zoom in and take a very close look at like 5 slides or something like that.
Okay.
Yeah, so they don't look at 1,000 slides and like aggregate all the information in their minds. It's usually just a sampling, just to see, because usually the errors that are made on one slide will typically be reproduced in the other slide.
So my other question is, sorry, don't worry. So on the public data set on TCGA, have you released these annotations?
Yes, so the heat maps are available upon request. They're not uploaded, but we are happy to, or at least when I was there, we agreed that we would give them out to anyone who asked, and we have given it out to a couple people. So you should email the corresponding author, and they should be able to give you the heat map data.
Okay, thank you.
Brilliant, thank you. There's just one other question from the chat from Craig Miles, who's asked, what zoom level were these patches extracted at? I'm assuming this is for training the CNNs.
So I think there were both 20x and 40x. And then if I'm not 100% confident on this, but I think we ended up just training with either 20 or 40.
Okay, brilliant, thank you.
Great. Any other questions on here before I move on?
I think we're okay for now, so yeah, feel free to carry on.
Great. So just to show some of the results, so this is the original H&E image. This is an example in stomach adenocarcinoma. And then this is the tissue prediction. So you can see red is the cancer tissue, orange is cancer-associated stroma. There's not really any necrosis, except maybe a touch in the blob sort of towards the right. And then these are the cell predictions, where you can see just like this river of lymphocytes and macrophages and fibroblasts and other things running in between the cancer epithelial cells. And then the red dots all punctuating the cancer tissue. And then this is just the overlay. And so this is generated, again, the tissue predictions are at 4 micron resolution, and then the cell predictions are 2 micron resolution, and the inference is performed on the entire slide. And then, so this was stomach adenocarcinoma, but of course, we did this across the 5 different cancer types, and you can see there's quite a bit of variation in the structure and in the properties of how the tissue organizes itself and how the cells organize themselves. I know these are like very small, not necessarily representative cuts, but I'll show a little more about how we can quantify these differences at scale in a bit. So that was step 1. So we got these heat maps. And so step 2 is to now extract these image features from the heat maps. So you start by getting the different predictions for all the different cell types and all the different tissue types. And then you can think about how these can be combined to form these different features. So, and then I attached a little image to the right of each of those, just trying to give like a visual understanding of what each of these are. So fractal dimension is pretty hard to understand, but it's essentially a measure of the complexity of the edge surface of the, in this case, excuse me, cancer-associated stroma. Eccentricity, a property of basically how elongated an ellipse is. The area of necrotic tissue, you can see there's a lot of black in that picture. Mean cluster size. I'll talk a little more about that in the next slides, actually, so I won't go too much into that. But essentially, the main takeaway here is the different tissue and cell types get combined for each of the for many of these. So it's not just number of fibroblasts or area of stroma, but things like the mean cluster size of fibroblasts in the stroma. And so this is motivated. I'll go quickly through this. Just some of the motivations of how we got the features that we ended up extracting. We just basically poured through the literature and found anything that we thought had been shown to be associated with properties of interest, like subtypes or which cancer it is or prognosis or anything like that. So weighing it all demonstrated there are a lot of different morphological features. Like in this case, you can see they're quantifying eccentricity and the perimeter to area ratio, and showing that can help predict prognosis. Lennon et al. Reviewed some work showing that texture properties like fractal dimension can be used to separate lung adenocarcinoma from lung squamous cell carcinoma. Saltz et al. Investigated the spatial relationship of clusters of tumor infiltrating lymphocytes and used that to predict certain features of the cancer. And then, so for this plot, I won't go into everything, but I thought this was an interesting way of trying to wrap your head around what these features mean. So essentially, the left-hand side is this histogram of the feature. So in this case, it's the density of lymphocytes within 80 microns of the cancer stroma interface. So at the top, you see there's a lot of lymphocytes in this cancer stroma interface, and in the bottom, there's very few. So those represent the range of possible values. And then here it's the mean cluster size of fibroblasts in the cancer-associated stroma. So you can see the top one has a big cluster, a lot of fibroblasts in it. Second one has smaller clusters of fibroblasts. The third one here, density of lymphocytes, the ratio of the density of lymphocytes to cancer cells in all of the cancer tissue, including stroma and tumor. And so essentially, I was sort of trying to bucket these as trying to draw exemplars of the way these HIFs can capture both count and density data, as well as cluster data, as well as things about relative proportions and proximity of cells to each other and cells to certain tissue regions. And then we have the tissue level features as well. So similarly, necrotic tissue, the top one, a lot of black you can see on the slide, bottom one, not so much. And there's a lot of different area and multiplicity HIFs, things about the architecture of the tumor. So in this case, you can look at the solidity of the tumor. So the top one, big block of red, pretty solid. Bottom one is much more interspersed. And so that can be captured by this measure. And then the perimeter square to area ratio captures the roughness of the edge of a certain region. So in the top, you can see the edges are of the necrosis are quite jagged and unstructured, whereas in the bottom, they're very smooth and neat. And so that's also something that we can quantify with the HIFs. Okay, I'll pause there if there are any questions.
Yeah, we've got a quick question from Nasir in the chat. It was, how many pathologists on average annotated at the cell level? And was there any, how many pathologists on average annotated at the cell level? And was there any QC of annotations on intra or inter-observer discordance between pathologists' annotations?
Yes, so I have, I'll show data on the comparison to interpathologist correlation later. So I have a slide on that. And then the first question was, how many pathologists? I can't remember, but it was sent to a network of hundreds of pathologists, and each job would just be, you know, whichever pathologist would sign up for it fastest. And then there were, you know, substantial variation in which pathologist would pick up which annotation job. And so it was from a pretty diverse set of pathologists. There was not any QC on those annotations.
Okay, thank you. We've also got another question here from Matteo Pazzi. And he asked, I did not completely get how you extracted the HIF from your model. Did you exploit the HIF from the pathologist's annotations or from features of the CNN?
Oh, not from any pathologist annotation, just from the heat maps output by the CNN. So, for example, the heat map will output this heat map, the model will output this heat map above, and then you can just take all the significant regions of necrosis, meaning any blob that's bigger than 10% of the largest blob, and then calculate its perimeter, calculate its area, take the square, take the ratio, and then just average all of those, or take the max or whatever it is, and that will be your feature. So essentially, it's the HIFs are computed just from the heat map and using only the heat map.
And just as a follow-up question there from, it's great. Did you try several different ways of obtaining the heat maps?
Sorry?
So just a follow-up question from Mattia here was, did you try several different ways of obtaining the heat maps? I don't know if you want to.
I'm not sure what you mean by other ways of obtaining the heat map. What would be another way of obtaining the heat map?
With GradCam, so using your CNNs and using GradCam on them.
Yeah, we did not play with other methods of obtaining the heat map. We relied just on the tissue and cell level models that were trained using the process described earlier.
Brilliant, thank you.
Great. So now we can talk about step 3, which is, and here it just says training ML models to find relationships with molecular information. But maybe before we do that, we can also just look at the HIFs that came out of step 2 and just their distribution and what they can tell us besides, and whether they're actually real. So this is the dimensionality reduction using UMAP of the different of all the different slides that we have. I can't see if the legend is, okay, it is. Yes, so they're colored by the cancer type, and you can see it stratifies quite well. So it seems like the HIFs are able to differentiate between the different cancer types. Interestingly, and not, I guess, not terribly surprisingly, the 2 types of lung tumor end up getting meshed together, but still with some separation. And then we also see that the different types of adenocarcinoma, so lung, stomach, and breast adenocarcinoma, tend to have a little more overlap than, for example, between squamous and other types of cancers. And then skin cutaneous melanoma also is the only one to have examples of metastases in there. So it would not be entirely surprising that it has a couple more points that are sort of broadly scattered across the space, whereas the other ones without metastases would be a little more consistent in their distribution of HIFs. And so just to quantify this a little bit, we can take the HIFs and cluster them. So we used a hierarchical agglomerative clustering method using the complete linkage method, and essentially clustered it into 34 different clusters of HIFs. And the reason we do that is because there's a lot of very high correlation between a lot of the variables. So for example, if you have like number of plasma cells and density of plasma cells and ratio of plasma cells to cancer cells, they're all going to be highly associated. So so we cluster them just to be able to summarize them without 607 rows. And then we pick a representative HIF from that list, but just the one that seemed to be the easiest to understand, that seemed to be most similar to all the others. And so in this case, you can see that, for example, skin cutaneous melanoma on the left tends to be enriched in a lot of the HIFs that are lower down on the list. The lung cancers tend to be enriched in the top 2, so macrophages and plasma cells and the cancer-associated stroma, and other features that you can use to essentially help determine at a very broad 60,000-foot view level what kind of features typically help differentiate the different cancer types that we're looking at here. And then the next thing we wanted to do is see whether this was associated with other things that we would expect it to be predictive of. So things like immune signatures. So in this case, we had different gene expression signatures from largely from the pan-immune data set, but also from other folks who went and analyzed and put up intermediate data files on TCGA. So what you see here is, for example, if you look at leukocyte infiltration and IgG expression, you'd expect these to be strongly associated with B cells. And in fact, we do see them most strongly associated with things like the cluster of HIFs correlated with the density of plasma cells in the cancer or density of lymphocytes in the cancer. So that reinforces what we would have expected. Similarly, if we look at the wound healing signature, the rightmost column there, we'll see that the amount of necrosis and the amount of plasma cells are all things that have been documented in the literature to be associated with wound healing processes. And then similarly, fibroblasts and stromal measures are associated with expression of TGF-beta, as well as density of fibroblasts in different parts of the stroma or in the tumor. And then in the middle plot, we aim to compare leukocyte fraction as estimated from our image. So essentially, like what fraction of the cells are leukocytes, white blood cells, versus the same estimate derived from bulk RNA sequencing. We got reasonable association there. It does undershoot the x equals y line, potentially because our model is only predicting between like 5 or 6 cell types rather than all the multitude of cell types out there. So we would expect it to undershoot over there. And then if you look at tumor infiltrating lymphocyte density, there was another study that collected many patches and annotated the ones that were enriched in TILs and used that as a measure of just how many, just the overall quantity of TILs in the tumor. And then we used our own measure, which is just lymphocyte density in the tumor, and found a reasonable association there. Although, of course, their method at the patch level, we expect to differ from our method, which is more exhaustive prediction. Oh, right, great. And then we get to the advertised prediction task, which is prediction of molecular phenotypes. We did predict the z-score of the PD-L1 or other immune signatures directly, but in order to produce ROC curves, we ended up binarizing the expression data. And we didn't want to cut at the 50% threshold because the clinically meaningful threshold might not be one that separates the population evenly. And so we ended up using this Gaussian mixture model analysis where we modeled it as 2 separate Gaussians, and then just wherever the density curves intersected is where we drew the split. And so for some of them, like the first one in BRCA, ends up being pretty close to the middle because it just looks like a normal distribution. And then for others, like for lung squamous cell carcinoma, it ended up drawing the line at where potentially there might be just a little bit of a divot, which ends up making it more of a 2-to-1 split instead of a 1-to-1 split. And then we also wanted to note that there's definitely some interdependency between the different 5 prediction tasks that we have. So HRD, or homologous recombination deficiency, is a little more independent. It's more of its own thing, but all the immune checkpoint inhibitors, all the immune checkpoint proteins end up being pretty closely associated with each other, as you might expect. And so it should not be surprised that if it does well on one, it'll do well on a lot of the others. So the way we did this prediction is we wanted to keep it simple with like a linear model, but we also wanted to account for the really extensive correlation between the different variables, the different covariates that we have in our HIFs. So one way to do so would be a group lasso, where you select for different groups. So maybe some clusters are just completely meaningless and we want to just drop them as a whole. But then it also means that, you know, we want to be able to drop within each cluster as well. So there's a method for that called sparse group lasso, which is essentially just a linear interpolation between the additional cost function for the group lasso and for just the vanilla lasso. And so that's something unfortunately requires like a good amount of hyperparameter tuning. And so the way we approach that is with nested cross-validation, where we took the, you know, we hide our test data, we bury it in the ground, we don't look at it, but we take our training data, our 80%, and then we want to split it into, we want to do one level of cross-validation in order to determine the hyperparameters, and then another level in order to get a sense of the variability of the model. And so that's why we have it nested this way. Yeah, happy to answer more questions about this otherwise, but I think it's fairly straightforward procedure. And then here's the outcome for PD1 and PDL1. These are the ROC curves. The overall splits, even though they're not guaranteed to be 50-50, are generally not that far off. So we felt that the ROC curve was appropriate, although we also do have PRC curves as well that I'm not going to show today, but are in the supplement of the paper. And then we were able to look at which features ended up being having the largest contribution towards the prediction by taking the ensembling the different beta coefficients that are produced at different parts of the cross-validation step, and then plotting them here. So you can see that the density of lymphocyte, or the cluster associated with density of lymphocytes near the cancer stroma interface, tends to have the highest beta coefficients and the greatest influence on the decision of whether PDL1 expression is high or not. And then for PDL1, it ends up being this exact same cluster, which is not surprising because they're fairly associated outcome variables. And then you can also look at the radar plots, which are more normalized against the maximum ensemble beta. And so you can see that a lot of the signal is primarily coming from lymphocytes, and then not just lymphocytes, but lymphocytes specifically around the cancer stromal interface. And then you can do similar analyses to break down where signal is coming from for prediction tasks involving the other immune signatures, and for CTLA4, for homologous recombination deficiency, and for TIGIT. Okay, so finally getting to that question that was asked earlier about comparison to interpathologist correlation. So this is internal. So this is just using validation data. And so what you see is that we generally tend to be around or within batting distance of interpathologist correlation, which we generally consider to be the best that a model trained on those annotations can do. And then just to validate this further, we also took 72 slides from an external data set of breast cancer adenocarcinoma. I'm trying to remember what the data set was called, but it's a public data set, and 72 H&E slides of breast cancer, and demonstrated something similar where I think we underperformed a bit in cancer cells, and did the same in lymphocytes, underperformed a bit in plasma cells, but did better in macrophages and fibroblasts, which doesn't necessarily mean we actually did better, but at least means that reasonably confident that those are getting pretty close to the best we can do. Okay, so that's all for results. Maybe I'll just power through just the limitations and takeaways, and then we can just do general questions after that. So there are a lot of limitations to the study, of course. So with the TCGA data set itself, we only have H&E stains. It'd be really great if we had like Ki-67 or immunohistochemistry. There'd be a lot of really cool other analyses we could do to either validate or to study other things that are in clinical practice, but alas, we do not. TCGA is also less likely to have samples, it has very few samples with metastases. The only cancer type that did was skin cutaneous melanoma, and not enough to do a separate stratified sub-analysis, although we did try. And then it's also underrepresented in samples that have more diagnostic uncertainty. And then just more limited data on treatment or clinical endpoints compared to data available through, for example, some of the commercial partners that we would work with. And limitations of the HIF method itself. I think the big one is generally requires annotation, and annotations are very costly. We had a lot of annotations, and that can be rate limiting. Morphologically similar cells can be easily confused by heat map models. We tried to fend this off as best as we could with input from our pathologists and targeted annotations, but there are still some that were very resilient to our efforts. So, for example, for stomach adenocarcinoma, when the tumor would infiltrate into the muscle lining, a lot of the smooth muscle cells got incorrectly classified as fibroblasts. And so that's something that potentially, if we had dedicated more resources and effort towards squashing that out and collecting more targeted annotations, we would have done a little better on that. But that's one limitation, is that oftentimes the class will not actually represent just the class itself, but rather a broader set of cells that are primarily that class, like primarily fibroblasts, but still have leakage from other things that kind of look like fibroblasts in the right setting. And then one other thing that we did not have time to improve was the HIFs are computed as slide level summary statistics. It would be very interesting, I think, to be able to break it down into the different parts of the slide and see how these HIFs differ across the image, or make sure that like a really important part of the slide doesn't get averaged out by the rest of the big slide. And so being able to account for spatial heterogeneity would be something that would be really great, but is not accounted for by this method. Yeah, so some quick takeaways. We think our data suggests that these HIFs that we've computed can capture canonical immune signatures, can recapitulate sequencing-based cell quantifications, can predict treatment-relevant molecular phenotypes, and can identify, you can trace back cell and tissue-specific contributors for future study and investigation. And so, as before, pros are interpretability, biological insights, limits of refitting. Cons are costly annotations, slow inference process, and the potential for underfitting. So huge thanks. I want to give a shout out to my partners in crime, Jason and Wan Feng, who co-led the study with me, and Dr. Rick Mitchell, who is my thesis advisor, who provided a lot of the guidance for what would be most clinically useful. To our pathologists, Dr. Kerner, Dr. Rao, and of course, PathAI CEO Andy Beck. And then my mentors, Dr. Elliott and Taylor Wiener, who provided amazing guidance on this project, and everyone else who supported the work. So that's all. Thank you so much for listening. It was a pleasure to chat with y'all.
Brilliant. Well, thank you very much. This was a great talk. And with that, I suppose I'll open the floor to questions. So actually, I can start. So, of course, you've looked at your nuclear segmentation, well, your nuclear detections. If you can instead change those to, say, nuclear segmentations, do you think that that would add useful information, or do you think it would get sort of lost in the fact it's static level summary features anyway?
That's a really good question. I feel that nuclear information would be helpful and would be interesting. I feel like it would add something. I feel like, again, there's the issue of, like you mentioned, not being able to account for heterogeneity is a limitation. But I think even with that, I'd be very interested to see a repeat analysis that also looked at things like the nuclei and even like nucleoli and blood vessels. And there are just so many other parts of the, I think that's one of the drawbacks of the HIF method, is that it can only learn and draw associations with what you have carved out for it. And so all these other really important biological entities that may play a role and mediate associations between the entities that we have annotated and the outcomes that we're interested in. Yeah, I'd be especially interested to see how those change when we add in information from those other entities.
Yeah, definitely. Might be a nice opportunity for your nucleic model, Mustafa. Oh, sorry.
Yeah, ask a question.
Yeah, go for it.
So thank you very much for the great talk, James. So I'll have you might have mentioned this and I missed it. So you already mentioned that you had trouble with annotating the macrophages, right? And also detecting them automatically because it's a hard task to do. How much does macrophage contribute to any of the predictions that you have done? Like, can you give me a summary? Like, is it a necessary thing to do in the experience that you had?
So I think so the reason why we poured so much effort into it was because we thought, oh, maybe it might be helpful. And it was doing pretty poorly, but there were just a lot of associations in the literature. And we felt like having it would be important. And we hypothesized that it might be important, but the thing is we froze the model before we did any of the downstream analyses. So if we had the chance to peek ahead and see the results, we would have seen that macrophages actually didn't have as much of an influence on the results as we had hoped. And maybe with the benefit of foresight, we would have chosen to invest that effort into something like nucleoli or blood vessels instead. But that's what we chose to go with. And if you just look at the some of these plots here, macrophages, you know, they had some signal, but it wasn't a dominant source of signal by any means.
Okay, and we are saying that lymphocyte is like pretty dominant here, right? So what kind of features related to lymphocytes did you find most useful? Like, was it the density of them or the distribution?
We think it's the distribution. I think, and it supports a lot of previous findings in the literature that lymphocytes in the tumor are very different from lymphocytes in the stroma, and their roles are different. And even breaking it down within B cells, plasma cells and lymphocytes function differently and mediate different processes. So yeah, I think in terms of further investigation of which areas would be most promising for further investigation for future therapeutic targets, or just understanding what would be most predictive for downstream tasks like prognosis or diagnosis or so on, my money would be on the B cells.
So actually, that you kind of answered my next question, was about other prediction tasks that you can do with the same data set. So like survival prediction on TCGA data set. Have you tried that to see what kind of features are important there?
For other prediction tasks like prognosis. Yeah. So we didn't get a chance to do that because I concluded the project, and I think I guess we didn't seek to do any of those other prediction tasks. But now that I think about it, it wouldn't be terribly difficult to just take the matrix of HIFs, which is pretty small, and associate that, just train a Cox model for prognosis. I'd be interested to see. I think we did a power analysis. Okay, I'm remembering that. We did a power analysis and it didn't look super promising. We were a little nervous about this idea that we didn't have treatment data, which we thought would be a huge confound that we couldn't control for. You know, if someone was taking a targeted therapy versus just getting like platinum therapy or general chemo. That would be something that would like throw a wrench into things. And we were only powered to discover hazard ratios of like up to, I think it was like up to like 1.5 in BRCA, which is the most powered set, or maybe it wasn't BRCA. I think BRCA was actually less powered because mortality rates are lower there. But essentially, we didn't feel that we were powered to discover the hazard ratios that that would be clinically meaningful, and we were worried about the confounding. But maybe we should have just given it a whirl anyway, just to see.
Yeah. So note that HIFs feature matrix is like small in size. Do you consider sharing it with the community at all?
Yes, the HIFs are actually public. So I think if you just
they
go
are?
to,
Okay.
yeah, they should be completely. So the heat maps are really large files, so I think you have to ask for that directly. But the HIFs, if you just go to the GitHub repo right there, you should be able to clone it and download and reproduce analysis.
Okay, that's great.
Brilliant, thank you. And we've also, Nasir said in the chat, great work and excellent talk, James. So thank you very much. I suppose one final question for me really is, so of course, one of the main benefits of these HIFs is that they are at the slide level and you've got 600 odd features that you can interpret quite nicely. I know you've already said that you potentially lose context by looking at them at the slide level. So did you think about, I don't know, tiling up the images and looking at tile level HIFs, for want of a better phrase?
Yeah,
So
I think so.
Sorry, go on.
Yeah, I agree. I think that would be the best way to do it. I think one question would be, how would they be tiled? I think one thought we had was organizing it around the different clusters. So for example, the clusters are already capturing some kind of heterogeneity, but only for certain features and only the cell level features. You could also just do it more naively, just by chopping it up into like a 3x3 grid or something like that. But yeah, I think we didn't get a chance to explore how we would generate tiles to have like a distribution of HIFs per slide. But I think that would, my suspicion is that would be an important step to ensure that it doesn't just get averaged out if it appears in one part of the image and not another. Yeah, things like that.
Brilliant. So I'll just see if there's any more questions from the chat at all. But if not, I suppose we can call the talk to a close. But that's been it's been brilliant. So thank you very much. It's been really interesting to hear you.
Thank you so much for having me.